All right, we're on.
Cool. All right. Thanks, Jen.
Sure.
Let's see. A couple things just before we get started. Today is just an overview. It's not going to take 2 hours. We'll really dig into the course material starting next week. Today's session is mainly just to introduce people to what the class is going to be all about, to see so that they know what they're in store for. It's going to be roughly a 20 to probably 25-hour commitment if you take the whole class. And I thought the other thing maybe I'll just say about this class is thanks everybody for coming. You guys are the intimate friends and family we were hoping to come to the first session. This is the first time we've ever put together this presentation, and what we're hoping is that you guys can be really blunt in your feedback. For instance, is this the right blend of clinical information versus informatics information? The way that the course is designed, it's roughly 75% clinical and about 25% informatics, with the thinking that people who are pursuing genetics informatics will benefit from understanding the clinical material. That's our opinion, but we're interested to know what your opinion is too. So, please think of yourselves as intimate friends and family. We're really looking for as much feedback as you're willing to give us on this, and in the process, we hope that we can import upon you the same, I guess, really wonder and marvel that we feel when we explore the clinical aspects of human genomics and human genetics. It's really such a marvelously interesting field, and we hope to kind of leave you after several of these sessions walking away just with your head in the clouds saying, oh my gosh, this is just unbelievable. That's one of the things we'd like to do. Because today's session is on the short side, it's just the overview, I was hoping we could go around the room and have each of you introduce yourselves and just speak for a minute about why you're here and what you hope to get out of the class. And I have a list of some of the participants here, and I'll just go down the list based on the alphabetical ordering here. And again, maybe you could just introduce yourself and talk a little bit about why you're here and what it is you hope to get out of the class. So, Anil?
Hi, I'm sorry, I was just trying to unmute myself. Can you hear me?
Okay. Yeah, welcome.
Yeah, thank you, Bob. So basically, I have been working with James Shalaby, PSMI, for the last couple of months. And the reason I'm here is that owing to my HL7 background, which makes me very comfortable in the structured data, there are some pipeline projects which are coming pertaining to clinical genomics as well, which is not my area of expertise. So I want to learn that and see how we can marry the structured clinical data versus the genomics data. That's what I want to learn.
Thank you.
Back to you.
Brett?
Thanks, Bob. Thanks, Jim. It's funny. I like the way you started, Bob. There's just the excitement around genomics and in the future. I feel like my experience is I'm comfortable with structured data also and moving data, but I have very little foundation in human genetics, and I know there's no greater person to communicate complex concepts than Bob, and so I appreciate you taking the time to put this together. I think looking forward to having a foundation at the end of this.
Oh, thanks, Brett. Denny?
Hi, sorry I disconnected and just got back with you. So I missed part of the question.
Main question, Denny, is if you could say why you're here and what you hope to get out of the class.
Well, my biggest problem with lab and LOINC and SNOMED has been with these types of records, and so I want to understand them better so I can do that work better, basically.
Great, thank you. Gay?
Hi there, Gay Dolin, and I'm with Intelligent Medical Objects. Just also wanting to get a better understanding of genetics and and how we can potentially do some innovative products surrounding that. As a side, I was, did 20 years in the NICU, and so had lots of exposure to really seeing, at least from the newborn side, the effects of some mutations and things.
Great, thanks. And Jonathan Gold?
Hey there. Hi, Bob. I actually took a course with you many years ago at FEMA.
That's right. Yeah.
Which I enjoyed tremendously, by the way. And I'm looking forward to learning more about genetics. It's one area within informatics I have not been following recently. And I work with Gay at IMO and would like to hear more about this and where we can go with that.
Great, thanks. Jonathan Lukoff on the line?
I am. Good morning.
Hey.
So I am a retired pediatrician who also did neonatology and obviously have some experience in clinical genetics because newborns sometimes look funny. Sometimes you take care of the sicker kids a lot in the hospital who have a lot of genetic problems. So I was a little more involved in some. And then I was walking down the hallway one day 20 years ago, and Bob Dolin said, "How'd you like to do clinical informatics with me in medical terminology?" So I got involved in that and a lot of work with SNOMED, and actually did some work in genetics in SNOMED, but probably not at this level. And about 3 years ago, I decided to attempt and succeeded at passing the boards in informatics. So I'm board certified in informatics as well, and Bob helped me study for that. So I'm here to learn some more from Bob.
Thanks. And Pale?
Yes. Hi, I work for Intelligent Medical Object, and during my master's, I took a class on bioinformatics where we learned a lot of tools and algorithms. And right now, with all the trends on precision medicine, I'm interested in application of informatics in genetics.
Thank you. Rafiq?
Hey, Bob. I got interested in genetics when my son was diagnosed with a genetical problem. So it always excited me to see how it works and how it can be leveraged for future predictive analytics. I'm also working on some of the machine learning, so it's always great to learn from you, Bob.
Thank you. Ron?
Hi. Sorry, I was on mute. Yes, Ron Dion here. I think several things are intriguing about what I hope you will be presenting. First of all, I just find myself, in general, way behind in my knowledge of genetic testing. And specifically, I've been encountering in several of my projects genetic testing, genomic testing, and all the new types of testing available, and the techniques, and I'd really like to gain a greater familiarity with the testing of those. And, then also the whole business around cytochrome P450 and how that type of testing is becoming available to better tailor drug choices. So all that kind of stuff.
Great. Thank you, Ron. We have somebody whose name is Viewer8.
What if that's me?
Well, Jim, you're on here too, but then there's a Viewer8, and then there's a boatload of people that are just on the phone.
Yeah, if they didn't put in their name, it when they joined, it would show up as viewer 1, 2, 3, 4, 5. Or if you joined using the the web, it'll duplicate your name. That's fine. But did we miss anyone? Did anybody want to speak up that's attending? And because we may not know your name from the screen. Yeah. I know there's somebody lurking in there. Carol, are you out there? There's Ev. Or trying to think who else. Adrian. No. Okay, then it must be just an echo.
Cool.
Yeah.
All right. So let's see now. You guys should be able to see the first slide, the cover slide, Introduction to Human Genetics.
Yep.
Right? Okay. And you'll see that there's a URL on this slide, and you can go there to download the PowerPoint. And this URL, we'll keep this URL for all of the course material. So right now you'll only find this PowerPoint up there, but as we move in, as we progress through the class, we'll continue to post all the material to this particular site.
Mm-hmm. And if you also happen to have a really strong firewall, some folks have problems with our URLs. Just drop us an email, and we'll maybe post it to Dropbox or or Google Drive. But if you otherwise, it should allow you to directly download it without a login.
Okay, and by the way, please feel free to interrupt, ask questions along the way. Today there's not a whole heck of a lot of material, but going forward there's a lot of material, and if you wait till the end, you might be a little bit overwhelming. So my preference is if you have questions, just speak up. There's not so much material that we don't have time to deal with questions along the way, and especially if it's going to make the difference of you following the discussion or not following the discussion, it'd be much better if you simply stop us and ask your questions. So today what we're going to do is walk through the course, talk about the different sessions. We're going to look at the calendar so you know when each session is going to be. Questions for the class. This is going to be your first homework assignment. There's 7 questions that I'm hoping each of you will, they're really brief, it'll take you like a minute or two, but I'm hoping that you guys will each answer those questions and send your responses back to me. It's mainly geared at assessing your baseline knowledge of genetics and better assessing your objectives for the class. So that we can do as good a job as possible tailoring the content to meet your needs, and also, tailor the content based on what your baseline understanding of genetics is. And then we're going to talk about homework. There are going to be 3 homework assignments during the course. They're not too onerous, and hopefully they're very fun because they're very informatics oriented. To really get your hands dirty with some of the informatics topics. Then there's some optional homework assignments we'll talk about. Then we'll talk a little bit about vocabulary. Genetics is a language unto itself, and so we're going to introduce some vocabulary today, but then each week we're also going to be introducing a few choice vocabulary words to try to, ease you into it. And then you'll see every week we're going to cover vocabulary, and there's going to be references as well. And that's what we'll do today. So by way of course overview, this is really designed to deepen the understanding of genetics for those wanting to delve deeper into clinical genetics informatics. And we're also going to be introducing clinical genomics informatics activities. We're really going to the class isn't designed to teach genetics informatics. It's designed to teach you, it's really, you still hear me?
Mm-hmm.
Yep.
Yep.
Okay, I heard a beep. I just wanted to make sure it wasn't me.
Somebody joining, yeah. Mm-hmm.
So the class is not really designed to teach genetics informatics. It's more designed to teach clinical genetics background material to those wanting to delve into informatics. That said, though, if we teach the clinical genetics and show informatics related material kind of in parallel, it probably will make everything come together a little bit better. So right now, I would say the mix is approximately 75% clinical and 25% informatics. There'll be a little bit of informatics weaved in through each of the topics, and then we'll have a couple of sessions that will be dedicated just to informatics. And through feedback, you guys can hopefully comment on, is that the right blend for you? The target audience is clinical and non-clinical informaticists with little to no prior genetics education who are planning to delve into clinical genetics informatics. So if you are an informaticist and you already have considerable genetics education, well, then of course be careful, because we're going to try to hit you up to co-teach the class at some point. So you're still welcome here, obviously, and we're always interested in finding people who may want to help teach this class going forward. The current format for this session is going to be a weekly 2-hour webinar, and there's going to be one topic per webinar, and I'll show you in a sec what the topics are. Down the road, we may tweak the format. It might be difficult for people to commit to Thursday 11 to 1 Pacific for the next 12 weeks. On the other hand, all the sessions are going to be recorded, and so people aren't necessarily obligated to do it in the format we're laying out here. But this is also because we're kind of building out and fleshing out the content as we're going through the class based on your feedback. And so in the future, we might try to do this in a little bit more compact fashion. But for now, it's going to be 2 hours every Thursday for the next 12 weeks or so. And I think, Jim, correct me if I'm wrong, but we'll also make the recordings available to folks?
Yep. We'll make them available in the same link, and be able to view them or download them, and probably I'll put them on a personal link in YouTube as well, just in case there's a firewall issue, and we'll send that out.
So the next slide I'm going to go over is the topics, and we're kind of sort of anticipating that each topic is going to take a 2-hour webinar. But again, that's why we're kind of doing this this session with you guys to see if that makes sense. But the way the topics are designed, they're each going to take about 2 hours. When we sent out the email invite, this list has changed slightly since then with the addition of the clinical genetics informatics session. Initially, when we conceived the class, it was only about 10% informatics, but we then said, why don't we bump up the informatics? So now we're going to have the clinical genetics informatics session is really going to be focused on clinical genetics informatics, such as HL7 standards, and the next generation sequencing variant discovery section is going to be largely on bioinformatics. What are all of the computer-related steps that go into next-generation sequencing, variant discovery, determining the clinical significance of variants. So sessions 9 and 11 are very informatics-ish, and then we're going to be weaving informatics topics throughout all of these. We'll be having, for instance, HL7 examples, fire examples, and things like that. So here's the topics. Next week, we'll begin with the real material, Introduction to DNA Genetics. And that class is meant to be, probably going to be a bit overwhelming because we're trying to expose you to a lot of material right off the bat, with the notion that repeated exposure to the same material over multiple topics will help make that material sink in. So we're really trying to introduce a broad number of components of DNA and genetics right off the bat, So this will be an introductory overview to the field. We'll talk a little bit about the structure of DNA. We'll introduce some additional vocabulary. There'll be some fun facts about DNA. And the fun facts to me are just kind of trying to whet your appetite for some of the marvels of DNA. And you hear some of these fun facts and it's almost incomprehensible when we talk about how big DNA is, when you realize how often DNA gets mutated. Then session number two is a little bit of a step back from DNA per se. We want to introduce the notion of the cell and the cell division. It's a little bit more of a basic biology class. It's important that you understand the cell, the nucleus, what a chromosome is, so that we can then talk about some of the pathogenic processes that affect DNA. So we'll talk about mitosis, we'll talk about meiosis, we'll talk about what are the different components of the cell, the mitochondria, the nucleus, the ribosomes, etc. Topic 3, we call that the central dogma of biology, and it's the process by which DNA is transcribed into RNA and then translated into protein. A lot of people may not be familiar actually with what a protein is, and so we'll try to describe what some of the molecules like proteins are. And then the normal process by which, 20 years ago when I was first, or 30 years ago when I was first introduced to biology, the central dogma of biology was the end-all be-all. DNA is transcribed into RNA and then it's translated into protein. We know now that several variations to this dogma exist. In fact, many of these variations are just so utterly fascinating that even the uncommon ones are kind of exciting to talk about because they kind of expose you to some of the mysteries and marvels of the human body. And so we'll talk about some of the variations, including epigenetics, alternate splicing, post-translational protein processing. Topic 4 is going to introduce the notion of genetic mutations and polymorphisms. It turns out, obviously, that no two humans are alike. Our DNA differs from each other by as much as 0.5% to 1%. And so we want to talk about what is it that's different between two people and what is it that's similar between two people. When you look at someone's DNA, how does it vary from a reference DNA? How does one person's DNA differ from another person's DNA? So we'll look at the different types of changes and variations that are seen in normal DNA and also the different types of changes that are seen that produce disease. The basis of the variation through meiosis, through mutation, will also be described. We're going to try to talk a little bit about the magnitude of the variation so that you understand, for instance, if I look at my DNA and I compare it against the NIH reference DNA, we're going to see 5 million differences. And understanding the magnitude of the differences will help you understand the complexity in reporting back to the patient. What does that mean? What does it mean that my DNA has 5 million variations from the reference DNA? And how do we grapple with that? And then that'll kind of set the stage for number 9, the next generation sequencing, where you sequence someone's DNA, you find that there's 5 million differences, and then you got to scratch your head and say, yikes, now what do I do with all of that information? Topic 5, we'll cover the genetic basis of inheritance and how alterations in one's DNA can be transmitted to their offspring. We'll talk about classic Mendelian inheritance patterns such as autosomal dominant, autosomal recessive, words you're probably familiar with from your high school days. But then we'll also talk about inheritance of more complex multi-gene disorders, such as the genetics of things like diabetes or the genetics of things like hypertension, where there aren't simple autosomal dominant and autosomal recessive patterns. And these complex multi-gene disorders are far and away more common than the simpler Mendelian genetic disorders. And so it's important that we leave you with a proper understanding that, the world of genetics doesn't revolve around the simple Mendelian disorders. There's this much broader notion of genetics that covers, all of these diseases that have a genetic component to them, such as hypertension, schizophrenia, congestive heart failure, etc. We'll introduce you to family trees and pedigree analysis as well, so that you can, for instance, begin to understand how you can look at a patient's family tree and try to assess, is the disease you're looking at genetic, and if so, what type of inheritance pattern, so that it begins to clue you in as to where you might look for the actual mutation that's causing what you're seeing manifest. Topic 6 will cover genetic disorders. We'll look at a couple of disorders in particular. We'll look at not just the disorders. We're going to be looking specifically at cystic fibrosis and type 1 diabetes. And what we're going to do here is not just explain the genetic biochemical basis for these disorders, but I thought here what we'll do is we'll walk you through the discovery method. How was it that scientists were able to determine the genetic basis for, say, cystic fibrosis? How was it that we were able to isolate the gene? How was it that we were able to isolate the specific mutation? For diabetes, which is a much more complicated disease where there's not a clear mutation, there's a whole host of genetic variants that are statistically associated with diabetes. We'll also take a look at how scientists are approaching that type of disease to figure out what genes to hone in on as possible causative mutations. And then the tremendous variability seen in humans sharing the same mutations will be described, and what that means is that you and I might each have the exact same mutation, and in you it might be manifest quite pronounced. In me it might be completely silent. And so we'll talk a little bit about this notion of penetrance and this notion of expressivity for genetic disorders. Topic 7 will cover cancer genetics, and we'll cover the genetic basis of cancer, this notion of oncogenes and tumor suppressor genes, but also we'll wind up delving into the cell cycle, so that we can explain what it is these mutations are really doing. It's, how these mutations are mucking with the cell regulatory cycle to result in uncontrolled cell growth. So that'll be topic 7. Then we saved some of these other topics for the end because they're a little bit more complicated, and to understand genetic testing and next generation sequencing and genetic engineering, we think that you'll be more able to understand these topics having been through the earlier topics. So when we get into genetic testing, what we're going to begin to talk about are the various types of genetic testing that can be performed. Targeted testing, such as going in and looking for a specific gene for a specific mutation, to other types of testing, such as analysis of one's entire genome. We'll talk about prenatal testing, paternity testing, and disease-specific testing. But what we want to try to do is leave you with a big picture overview understanding of, this is what I'm looking for, this is the type of test that's going to be helpful, whereas if this is what I'm looking for, these are the other types of tests that might be helpful. So really help you understand the range of test types that are out there. Topic 9 will be on next generation sequencing, variant discovery. And I hope you find this to be a fun class because honestly, I had such a fun time putting it together. We're going to present a case. The patient's going to come in, and the provider is going to be convinced that the patient has a genetic disease, but on initial testing, that the genetic testing is going to come back negative. And so the provider is then going to have to perform next generation sequencing, and he's going to find that there's 5 million variants, and the provider is then going to have to comb through those 5 million variants to see if in fact there is a causative variant that can be identified. And so really this is an exercise in helping you understand the bioinformatics workflow that leads from next generation sequencing to variant discovery. So we'll dive into the actual next generation sequencing technology because it's important to understand the wrinkles and the bumps. Next generation sequencing technology doesn't leave you with this perfect string of 3 billion base pairs. It has considerable QA considerations, it has considerable workflow considerations, and it's important to understand the strengths and the weaknesses of next generation sequencing and how you can use that information in a bioinformatics pathway to ultimately try to determine, what, if any, pathologic variants might actually be present in the patient, that you're interacting with. So I hope that's going to be a very fun session. Genetic engineering is also probably going to be what a lot of people get excited about, and that's an overview of how genetic engineering is being used today, such as for targeted gene therapy or envisioned for the future, such as in stem cell modification. And I almost should change this because even though it's envisioned for the future, it's not as though research isn't being done in stem cell modification today. We'll talk about CRISPR-Cas9 technology, and we'll also talk about other tools that are being used for genetic engineering that allow folks, scientists, to alter your genes, reintroduce these genetic modifications back into the human to affect change. Topic 11, clinical genetics informatics, is going to be an introductory overview of the bioinformatics resources and the clinical informatics standards. The clinical informatics standards are primarily going to be the various HL7 standards, the V2, the CDA, and the FIRE resources, whereas the bioinformatics resources, these are going to be things, primarily the public knowledge sources, where clinical variants are housed at the NIH, for instance, or where you can go to look up the significance of a particular variant. So it's the genomic bioinformatics. And in the literature, informatics and bioinformatics sometimes are a little bit blurry. Here, when I use the word bioinformatics, I'm primarily talking about not the communication standards, but the computer tools that geneticists use to analyze the vast amounts of genetic information. So that's what I'm referring to as bioinformatics. And when I talk about informatics, I'm mainly going to be referring to the types of interoperability standards that we've been dealing with for a long time. Primarily the SNOMED stuff or the HL7 stuff. And so in this session, we're going to provide an introductory overview to kind of both of those domains. And then finally, topic 12 is going to be a chance for class participation, and it's going to be debates. And we're going to have a couple of ethical topics, and I'm going to ask you guys to prepare ahead of time. And then this particular class is going to be a debate. It's not designed so that people try to win the debate. It's really designed to help surface pros and cons so that each of us are then better informed and better able to make our own decisions when we're faced with these ethical dilemmas down the road. So I think number 12, many people find that class to be the most interesting because it's really just going to be a session where we can talk and debate and try to educate ourselves about the pros and cons of a lot of these different issues. So before I move on, any questions about the topics?
This is Gay. I do have one question.
Yeah.
Really basic, probably, but will you be talking a little bit about what's the difference between genetics and genomics, and when and where is the right proper way to use those words, and basically what the difference is?
Yes, I could. It's blurry, but mainly the main distinction I've seen is that when people talk about genetics, they're talking about DNA. When people talk about genomics, they're talking more broadly. They're talking about epigenetics. They're talking about the RNA. They're talking about the protein translation process. They're talking about the proteome. So I'll verify that, but my understanding is, genetics is DNA, genomics is much broader. And so the trend is, we used to talk about genetics because we thought the whole world revolved around DNA. Now we know that DNA gives us but a tiny glimpse, and that really genomics and proteomics and the metabolome and all that stuff play humongous roles. And so the trend is to move away from the term genetics and move towards the term genomics. Maybe we'll have to rename the class. Because we'll definitely be more covering the broader notion of genomics in the class.
Oh, thanks.
Any other questions before we move on? Okay.
Hey, Bob, this is Ron. You mentioned something about that people really enjoyed that last class, which seemed to imply that you've done this course before, or perhaps other courses have had the debate section. Could you clarify?
We've done a couple of very, very, very limited, a couple of these classes to a very tight group of friends. But this was during the development process of the class. So we've presented all of this material a couple of times before, just as part of preparing the material. But this is the first time we've really pulled everything together into like a cohesive class.
Thanks.
Mm-hmm. So you haven't missed anything. Okay. Calendar. Here we go. It's quite a commitment. When I look at this, I say it's quite a commitment. And obviously, when I look at the people who are on this call, I can't imagine any of you are going to be available every single Thursday for the next 12 weeks. But the sessions will be recorded. And this is our current plan. I think in the future, maybe we'll try to bunch them all up. It's hard to say. The material, sometimes you just need a couple of days to clear your head after you learn a little bit about genetics, and trying to cram all this material, for instance, into a one-week course might be overwhelming. So we'll have to continue to think through what the optimal timing is. But currently, what we're envisioning is one topic every Thursday, and it'll always be on this line. It'll always be on this website, and you can always access the material through that same URL. Here's the home. This is something that I would like each of you to do. Hopefully it won't take more than about 5 minutes. It's just download the slides, answer these 7 questions, and send me an email. I want to make sure that the class covers your objectives. And so I'd like you to just, if you could, a sentence or two. Why are you in the class? What is it you're hoping to get out of the class? Is there anything in particular you were hoping to get out of the class? If we see that there's people that are looking for specific things and it's not currently covered, we'll have time to make changes in the classes coming. It'd be helpful to know a little bit about your biology background. For instance, when did you last have a biology class? Do you know what is a cell? Did you already know that a cell has a nucleus? Did you already know that the nucleus of a cell contains DNA and chromosomes? Did you know that there's 4 DNA bases? Did you ever see the movie Gattaca, by the way? Any of you ever see the movie Gattaca? No? Okay, Gattaca was one of these popular movies about one of these new world orders where they can actually alter your DNA. And it was kind of clever because the title Gattaca was Gattaca. Anyhow. And then did you know that the DNA instructs the cell on how to make the protein? So if you guys could answer these questions and send me a note, it'll help me gauge, how quick or how slow we need to go as we proceed through some of the topics. And I would really appreciate it, so thanks. Okay. And this, I know, is probably most of your favorite slides, right? The homework assignments. Nothing to do yet, but this is just a heads up on, these will be the homework assignments that you'll get as part of the class. We'll ask you to build, and this can be a fake family tree. I'm not asking you to divulge any true health history, but we'll ask you to build a 3-generation family tree, including at least one condition. I'm still trying to explore what the tooling support is going to be. So the homework assignment isn't yet quite finalized, but we may not only have you build the family tree, but also convert it into an HL7 V3 representation, the kind that is required under meaningful use, and also a fire representation. So that'll be coming up in a few weeks. The other homework assignment, which is going to be closer to the end of the class, is I'm going to give each of you a DNA variant, and you'll have to poke around and try to determine the clinical significance of that variant. Some of the variants will be obviously clinically significant. Some of the variants, in the other extreme, you're going to have a heck of a hard time even finding anyone who's ever identified them before. Some variants you're going to find, but it's going to be a little bit tricky to know whether or not they're clinically significant or not. And that the purpose for this homework assignment is in tracking down your particular DNA variant. I hope it's going to make you feel a little bit more comfortable with some of the main bioinformatics resources that are available. The other homework assignment is going to be to prep for participation in the ethics debate. There's going to be 2 questions we're going to be debating, and in a few weeks I'll tell you what those questions are, so that over the course of the next month or 2, you can begin thinking about those debate questions, begin thinking about pros and cons, so that when the debate actually rolls around, we can try to better educate each other about the pros and cons of the various questions. Then we have an optional homework assignment. Oftentimes you're reading a magazine, and these days it's pretty hard to be reading a magazine and not see something related to genetics. And if you'd like, if you find an article, whether it's in a scientific journal or whether it's in a more of a journal that's geared towards non-clinical, like Scientific American or something like that. If you find an article that you think is relevant and timely, and you'd like to do some optional work, if you could write it up briefly, just say where you got it, give a 1-paragraph summary, and then if asked, would you be willing to give a 5-minute presentation on the article to the class? And this allows you to kind of get your feet wet, build a little confidence in your own genetics knowledge, because you would be presenting that article to the class. So if you'd like to do that, please go ahead. And if you have any questions about that, you can let me or Jim know as well. And we're getting near the end here. Every week, we're going to be introducing some vocabulary. This is a starter set of vocabulary. The actual source for this particular vocabulary, you'll see it in the PowerPoint. This came from a New England Journal article back in 2010. And this is a decent starter set of vocabulary, but there's just hundreds and hundreds of new words you're going to be exposed to. And so if you'd like, go ahead and peruse this particular list. And then what we're going to do each week is have 3 or 4 vocabulary words that we'll focus on a little bit so as not to overwhelm you. So this was a good set of words, and by the end of the course, I hope you understand all of these words at a minimum. Probably you'll wind up learning at least 2 or 3 times this many vocabulary words, but this is a good starter set, and then we'll dig into additional vocabulary each week. And finally, each week we're going to have, in addition to vocabulary slides, we're going to have a references slide. But I'm not going to list references twice. And so, for instance, the HL7 standards are listed here. I'm not going to keep listing them each week. And much of the material that's going to be presented is going to be pictures taken from these 3 books. So probably 80% of the figures that are going to be coming up are from these 3 books, a couple of medical genetics books, and this is this really, really fantastic bioinformatics book. This is really not an informatics book. For instance, this book doesn't mention HL7, doesn't mention fire. I know, hard to believe, but it doesn't. It's really focused on bioinformatics. What are all the computer tools that are out there for assessing the whole field of, and this is genomics, not genetics, so this is on, proteomics tools and RNA tools, and a really wonderful book. And we'll be using a lot of content from this book, in particular when we talk about some of the bioinformatics topics. And that's it. That's it for today. That's it for today, unless you guys have any questions.
And just wanted to mention, Gay had a kind of a good idea that I'll go ahead and send the questions that Bob, you were asking folks to fill out and just send back to you, so you can, hone the the presentations to the audience. So I'll just respond to everybody with those questions in an email, so that you'll all have it in an email form.
Oh, thank you, Jim.
Jim, could you send out the website address on that? I couldn't copy it from the picture on the screen, so I don't have it.
Oh, sure. Yeah, I'd be happy to. So I'll do a full response to everybody on that, and I'll also attach the deck, in that response, just in case you have any problems getting into the site. And if you just let me know, just send me an email if you run into a problem with that. We'll post it as well into a like a Google Drive site if we run into problems. Yeah. You shouldn't, but your firewall sometimes can do funny things. And you'll also get a link to the video. So that should, usually I'll just post those the next day, so you'll have those as well. Any questions, or all good?
Well, with the free hour you guys have, I hope you take 10 minutes of it to answer those questions and get them back to me while you still have time.
Yeah. And I'll send them out right now.
All right. Thanks everybody.
Everyone.
Talk to you guys
Thank you.
next week. Okay.
Thank you.
Thank you.
Thanks.
Thank you, Bob.
Thank you.